All right, so now we're back with part 2 of our brief review over cells. And so we're going to talk in a little bit more detail about some of these organelles. And we're going to start with the nucleus. And so the nucleus is, of course, it's the biggest organelle in the cell. It's easiest to see a lot of times. And of course, it contains the chromosomes. And it also contains the nucleolus. And the nucleolus are where ribosomes are synthesized. And so again, this is just a little diagram. You can look at the different parts here and see how they relate. But when we talk about protein synthesis later in the semester, we'll of course talk about the nucleus and the nucleolus quite a bit more. The DNA, of course, is stored in discrete pieces named chromosomes, and every species has a stereotypical number of chromosomes in its nucleus. And so each chromosome is just one single long strand of DNA. So remember when we talked about nucleic acids, and they're just long strands of nucleotides, and one long strand, that would be a chromosome. Now, technically a chromosome has a lot of protein that's used to help kind of wrap it up and give it a typical shape. And so you have the DNA, and then when it gets wrapped up with these proteins, that's technically a chromosome, but we'll use these two interchangeably. But again, remember that DNA and nucleic acids, they're not branched. They don't have pieces going off. It's one long chain of nucleotides holding hands. So again, our mantra, DNA makes RNA, makes protein. Again, every night before you go to bed, I want you to say this to yourself. So the DNA codes for the RNA. The RNA uses ribosomes to make the protein. So that's another organelle that we need to talk about. Ribosomes are made of RNA. And those ribosomes are synthesized in the nucleolus, which is within the nucleus. And so we'll talk about this again in more detail throughout the semester, but there's different types of RNA. And so there's rRNA, or ribosomal RNA. That's RNA that's used to make the ribosomes. And then the ribosomes go out into the cell, and then they read mRNA. So the ribosome is made of one type of RNA, and it reads a different type of RNA when it's trying to make the protein. Third figure from your book, just kind of showing you a microscope view of different parts of the cell, and you can see that inside the nucleus you've got the nucleolus where the ribosomes are synthesized. The little red arrows are showing some of the pores. Things have to get into and out of the nucleus, and so there's little holes for that to happen. Like we said, the ribosomes read mRNA, and that tells them what order to put amino acids together to make a polypeptide, which is going to be a functioning protein. And so since the DNA codes for the RNA, and then the RNA codes for the amino acids, that's how DNA makes RNA makes protein. So those ribosomes are going to make the protein in a couple of locations. And so there are free ribosomes that just float around in the cytoplasm outside the nucleus, and they just grab a piece of mRNA and translate it and make your protein. But there's also something called rough endoplasmic reticulum, or rough ER, and that's where the ribosomes are bound ribosomes, and they're bound to the endoplasmic reticulum, and they also make protein there. And so there's a figure from your book just kind of showing some of those details, and again a micrograph of what that would look like. And so here is just a kind of a drawing of a ribosome, and there's a large subunit and a small subunit, and these two are assembled around a piece of mRNA, and then they can translate that mRNA. And it's showing you also just under the microscope what free ribosomes look like and what bound ribosomes look like. And so when we talk about endoplasmic reticulum, that's part of the endomembrane system. So let's talk about that briefly. Endo means in or inside, And so the endomembrane system are the inside membranes. So we talked about the membrane that forms the outside of the cell, but these are membranes inside the cell. But they're made of the same stuff. And so some of the things that we need to know about the endomembrane system are the endoplasmic reticulum, the Golgi apparatus, lysosomes and vacuoles. Again, this is just a brief 10,000-foot view of the cell. Here's a figure from your book showing kind of the relationship between all these different organelles within the cell. Note again how they're all made of the same type of phospholipid bilayer, the same type of membrane. And so that's important because it means you can exchange pieces between them. And so you can have a piece of the endoplasmic reticulum bud off and form a vesicle, and then that vesicle can fuse with the Golgi apparatus because it's all sort of the same thing. So, you know, if you've ever played with bubbles or seen, you know, bubbles in the bubble bath or something, and you see bubbles and then bubbles come together and they fuse into a bigger bubble, that's the exact same thing, and that's what's going on here. It's because they're made of the same material. And then you can have something bud off of the Golgi apparatus, and it can go and bud to the plasma membrane and fuse with it. Or you can have different vesicles that fuse together. They can do this because they're all made of the same stuff. So in the ER, we mentioned this already. There's two distinct regions, the rough ER and the smooth ER. The difference is the rough ER has ribosomes. And so, again, just kind of showing a general diagram of these things. And this is not cell bio, so we're not going to get into tremendous detail, but you know, the smooth ER in general synthesizes lipids. It's where carbohydrates are broken down. It's where drugs are detoxified, things like that. The rough ER, of course, since it has ribosomes, that's where proteins are being synthesized. It's also where the membranes are assembled for the cell. And so that's what we're kind of just showing their relationship here. So the Golgi apparatus takes different vesicles of different things and combines them in different ways and processes them and then releases them so that they can go do whatever job they have to do. So, you know, you've got pre-products that come in from one side of the Golgi apparatus, and then the Golgi apparatus modifies them and releases the final product, and then that final product can go do whatever it needs to do in the cell. And so it modifies and packages these things. It takes things in from the cis face. Within the Golgi apparatus, you do the modification, and then it releases the final product from the trans face. And so, for example, you know, the rough ER, you're synthesizing polypeptides, and so maybe they're not quite the finished protein product. Remember, a polypeptide is not necessarily a protein. A protein is a polypeptide that has function. And so, for example, talk about things like proteins that have quaternary structure, you know, like a collagen fiber or a hemoglobin or something, where you need different polypeptides to all come together to form the functioning protein. Well, so a lot of times it's the Golgi apparatus that's pulling these things together. And so if that protein is synthesized at the rough ER, then you can have a piece of rough ER, you know, bud off and carry that polypeptide to the Golgi apparatus, fuse with it, and then release that polypeptide into the Golgi apparatus. And then, you know, the Golgi apparatus can modify it and then release it from the trans face. And so that's what we're showing you here, you know, and you've got vesicles coming into the cis face and leaving the trans face. And so again, figure from your book, just kind of showing the general view of this organelle. Okay, another component of the endomembrane system are lysosomes. This is just, again, a membrane, but within that membrane you've got digestive enzymes. And so this lysosome can go and fuse with different things and release those digestive enzymes, and they can break apart whatever needs to be digested. And so that's what they're showing you here. So, you know, as you take in food, that food gets broken down smaller and smaller pieces until those pieces are small enough to be taken into a cell by a vesicle. But then they still need to be broken down even further. And so once they get into the cell, the lysosome will fuse with that food vesicle, that food vacuole, release those digestive enzymes, which can do the final steps of digestion on that food. Another reason that we do it this way, you can't have digestive enzymes just bebopping around inside your cell, right? The digestive enzymes break down organic molecules. And so if they were allowed to roam freely in your cell, well, they would just break down all the stuff that's inside your cell. So they're contained in this lysosome. And so what we're showing you here is that lysosome fusing with the food vacuole and doing, completing the final digestion of food and preparing that then to go to the mitochondria or wherever it needs to go to be used by your cell. And now sometimes we do want to break down parts of your cell, old worn out organelles that aren't functioning anymore. Another use of lysosomes is that they fuse with a vacuole that contains these old organelles, and they break those old organelles down, and then you can reassemble the pieces back into a new, fresh organelle. The final piece of the endomembrane system that we mentioned is our vacuoles. And we just talked about food vacuoles. Plant cells or fungal cells also have central vacuoles. And these are great big vacuoles, and a lot of times they're it's just the same thing, it's just bigger. And it's a very conspicuous component of a plant cell is this central vacuole, which holds a lot of water. And so this is what allows cells to stand up, right? You got this big vacuole that gets filled with water that expands out against that tough cell wall, and it gives rigidity or turgidity to that cell. And if you got all your cells are very turgid, then they're very stiff and the plant can stand up. And that's why if you don't water a plant, these vacuoles shrink, and so they don't exert as much pressure inside the cell, and so the cell is not as turgid and it shrinks and the plant wilts. And so here's another diagram of that large central vacuole that you're going to find in plant cells but not in animal cells. And so again, just a series of diagrams showing the relationship and how things can move from one piece to another. And you see that things that can bud off from the endoplasmic reticulum, they move to the cis face of the Golgi apparatus where they can fuse because it's made of the same material. Then they can bud off from the trans face. The final products are in little vesicles that bud off from the trans face, and they can either go fuse with lysosomes, or they can go somewhere else in the cell, or they can fuse with the outer membrane and release the contents to the outside of the cell. And so all these components are easily interchangeable. Okay, next organelles we need to talk about are the mitochondria and the chloroplasts. They're very similar to each other. They're important because they change energy from one form to another. So we mentioned that all life, of course, is dependent upon a constant input of energy from the sun. That energy has to be captured by the chloroplast and turned from light energy into chemical energy. And then that chemical energy needs to be, bonds need to be broken and that energy released and then captured into other forms of chemical energy. And so that's what the mitochondria do. And that's the basic component of all living things, is transferring this energy from one form to another. And so we're going to talk about cellular respiration in more detail a little bit later, but this all occurs at the mitochondria. And so that's where we use where we use oxygen to help break these bonds and release their energy and put that energy into other molecules, specifically into a molecule called ATP, and that ATP then can take that energy and move it to other parts of the cell, and then we can use that energy to do other cellular processes. So we'll talk about this in more detail, Bottom line, ATP is like the cell batteries, right? ATP just takes the energy from your food and carries it to somewhere else where you need to use that energy. Of course, the chloroplasts are in photosynthetic organisms, plants, and algae. There are photosynthetic bacteria, but they don't have chloroplasts. They're prokaryotes. But this is where you take, again, you're changing energy from one form to another. You're capturing light energy and you're turning it into chemical energy. What's interesting about mitochondria and chloroplasts, we talk about them together not only because they're the sites where energy is transformed, but they both have similarities with bacteria. So for example, these are organelles, but they have their own ribosomes. They also have their own chromosomes. So you have two types of chromosomes in your cells. Your chromosomes that are in the nucleus, which are linear, but within these organelles inside your cell, they also have their own DNA, and it's circular, just like prokaryotic DNA. That's interesting. They also kind of grow on their own and reproduce somewhat on their own. So these are organelles that aren't, they can't live outside your cell. They're very important to your cell, but they look a lot like bacteria. So that's interesting and weird. What does it mean? Well, this is evidence for something called the endo-symbiont theory. And so endo again means inside. Symbiont or symbiosis means two organisms that live in that interact in close proximity. And so the idea is that an early ancestor of eukaryotic cells engulfed a prokaryotic cell. And that prokaryotic cell was a non-photosynthetic prokaryotic cell, but when this early ancestor of eukaryotic cells engulfed that bacterial cell, that prokaryotic cell, they both benefited. It was a symbiotic relationship, which of course we see all of biology. You see tons of symbiotic relationships. Well, this is a symbiotic relationship in which both organisms benefited. And so you've got these two different cells that became a single cell. And so over time, that cell that got engulfed could no longer exist on its own, and it just became an organelle. And so this is how eukaryotic cells evolved, is that they've got this new cellular form that's got this mitochondria. And then in another lineage of these cells engulfed a different prokaryote that was photosynthetic and gave those cells the ability to photosynthesize, and the same thing happened. It was a symbiotic relationship between that eukaryotic cell and that prokaryotic cell, and eventually the prokaryotic cell just became an organelle of the eukaryotic cell. And that's how chloroplasts evolved. So it's a very interesting origination, but it's interesting to think that all your cells, actually, the organelles are, their ancestors were bacteria. Okay, so if you can't hear that, it's a little distracting, but my ducks have shown up. Let me show them to you. And so there are my ducks. Hi guys. But you can see that their food pan is right behind me. And so they've come up to ask to be fed. And so I should have known this was going to happen sooner or later. Here's the thing. They're not going to quit. They're going to keep making noise until I feed them. So let's go ahead. I'm going to take a time out, and I'm going to pause the recording, and I'm going to feed these ducks, and then I'll be right back. Okay, I'm back. I fed them. So you might still hear them making a little bit of noise as they eat, but I promise you it's much less noise than they would have if I hadn't have fed them. You might be wondering why I'm so just dead set on recording all these outside when there's so many distractions, but if I was inside, the dogs would be barking, so I would just rather be outside, and it's worth it. So let's get back to talking about our cells, and we're talking about the endosymbiont theory. So again, just to summarize, early life, the first life that originated on Earth were prokaryotes, bacteria. And in fact, most life, if you look at the history of life on Earth, most of our time, of the time on Earth that things have been alive, it's been just bacteria. We eukaryotes and multicellular organisms have not been around very long relative to the age of the Earth and how long living things have been on Earth. So the prokaryotes were around for a long time, but at some point, larger, more complex cells evolved and became single-celled eukaryotes. But what allowed them to evolve was when one prokaryotic cell engulfed another one, and the symbiotic relationship between those two made the cells, allowed them to get bigger and more complex. And over time, those cells that got engulfed became mitochondria and chloroplasts. And then, again, single-celled eukaryotes were around for a long time, and then only very recently in evolutionary time did those single-celled eukaryotic organisms evolve into multi-celled eukaryotic organisms, which eventually led to us. So we multicellular eukaryotes have not been around. We've been around for a very long time in terms of years, but in terms of, how old the Earth is, we haven't been around very long. Okay, so this is just another diagram, just sort of underscoring and relating this idea of the endosymbiotic origin of mitochondria and chloroplasts. And so here's a figure from your book just showing the structure of a mitochondrion. Like I said, we're going to talk about cellular respiration in more detail here in an upcoming lecture, and so we're going to, the structure of this organelle is going to become very important. And just another diagram showing the structure of this organelle. Here's a diagram showing the structure and the location of chloroplasts. Okay, finally, the last thing we want to talk about is the cytoskeleton. The cytoskeleton is just sort of like the scaffolding within the cell. It's just organelles or structures within the cell that help to anchor other organelles and give an inside scaffolding to the entire cell. And it allows for things to move around the cell. So most stuff doesn't move around the cell at random. It actually kind of slides along this cytoskeleton. And there's three types of fibers in the cytoskeleton. The microtubules are the biggest. The microfilaments, these are also called actin filaments, and those are the thinnest. Now these are going to become important when we talk about muscle contractions. And intermediate filaments are intermediate to the two. So it's a very clever name. And so we're not going to get into too much detail. Well, these things will all pop up later when we talk about different things like the cell cycle and DNA being pulled apart and things like that. Okay, so that's just, again, the 10,000-foot view, a brief review of cellular biology. Let me know if you got any questions, and I'll talk to you later. Have a good one.